After watching this video, you should be able to describe the general mechanism underlying the megaloblastic anemias. You also should be able to describe the clinically relevant biochemical reactions involving vitamin B12 and folate and use that as a model to explain the various causes of megaloblastic anemias. You also should be able to list the major hematological findings seen in megaloblastic anemia and describe specifically the unique findings that are seen in B12 deficiency. So the major underpinning for megaloblastic anemias is that there's a problem making DNA. Okay, so it's an important place to start is for the general mechanism is that there's an impairment of DNA. So over here, say sort of general mechanism. Decrease in DNA synthesis. And what ends up happening when this occurs is that there's a decrease in nuclear divisions. The cytoplasm still matures normally, but they're not synchronized, and you ultimately end up with what's called ineffective hematopoiesis. Okay, so megaloblastic anemia is a very classic example of ineffective hematopoiesis, and the general idea is that there's a decrease in DNA. Now, if you recall, the normal development in the bone marrow requires lots of cell divisions. The cells start out big, and as they develop, they get smaller. And when this occurs, when you have this nuclear cytoplasmic dyssynchrony, it's really a maturation defect that results in the cells staying big. They don't develop well. In fact, there's a lot of intramedullary hemolysis, destruction of these large immature red blood cells before they even get out of the marrow. And the ones that do end up in the marrow are too big. Okay, and you end up with an increase in average red blood cell size. Now we're talking about the decrease in DNA. We might want to go a little bit more in depth on how that might work, because the two most common causes of megaloblastic anemia are vitamin B12 and folate deficiency. So it's a good idea to go and take a look at how vitamin B12 and folate are involved in that process. Now what this is showing here is that we have our DNA, and remember DNA requires deoxytrinucleotides, okay, and so this is deoxy-TTP here, for example, and that comes from actually deoxy-TMP, deoxythymidine monophosphate. And the way this works is during a synthetic pathway, you don't make deoxy-TMP directly. It actually comes from deoxy-UMP. You have a uridine monophosphate here, and that needs to get a methyl group to become deoxy-TMP so you can make this particular deoxytrinucleotide. Now, folate is a very important vitamin because it does a lot of carbon transfers. And the form of folate that's involved in this enzymatic step that's done by thymidylate synthase is the N5-N10 methylene tetrahydrofolate. All right, so this is what's going to donate that methyl group to this uracil molecule and make a T. Okay, and during this process, you form a dihydrofolate, and that dihydrofolate then has to eventually go back, finish the cycle, and make your N5-N10 methylene tetrahydrofolate. And the enzyme that's responsible for the DHF to THF step is dihydrofolate reductase. And you make your THF, and then the THF can get converted to your N5-N10 methylene tetrahydrofolate, and then you have a little cycle. Okay. Now there's other enzymes here that I'm leaving out the names for. The reason why these two are important, thymidylate synthase and dihydrofolate reductase, is that these are drug targets. So that has some medical relevance. Now, the N5-N10 methylene tetrahydrofolate as it cycles around, some of it gets siphoned off and forms N5-methyl tetrahydrofolate. And this form needs to go back to THF to participate again in the cycle. Okay, so think of it as like a little detour. And the way it gets back into the cycle is B12, vitamin B12. And another name for vitamin B12 is cobalamin because it has a cobalt atom. So cobalamin and vitamin B12 are interchangeable terms. At any rate, the vitamin B12 or the cobalamin picks up the methyl group, okay, from N5-methyl tetrahydrofolate and becomes methylcobalamin, and it regenerates the THF so it can participate in this cycle again and make your DNA. Now the methylcobalamin has to go back to cobalamin, all right? It can't stay in that form, otherwise things will back up. And it hands its methyl group off to a molecule called homocysteine. And when it gets methylated, it becomes methionine, which has some important roles itself, methyl donation to various things. And that goes off and does whatever it's going to do. So you can see that B12 and folate are intimately connected to keep this cycle going so you can make your deoxy-TTP. Okay, so I think it's obvious that if we had a deficiency in folic acid, okay, so for example, if you're thinking about dietary sources of folate, you know, for example, let's say your diet is coming in over here, folate in the diet, all right. If you had a problem with your folate, either you had a dietary deficiency or you weren't absorbing it, this cycle would be depleted and you wouldn't make the thymidylate synthase wouldn't be able to do its thing and you wouldn't make DNA, and then you'd end up with this megaloblastic anemia, all right? So you can have a problem with the diet, a problem with absorption, right? As a couple of examples for folate deficiency. Now, what if you had a B12 deficiency? You know, we also have B12 in the diet, you know, comes in there. Now, it's a lot harder to get a dietary deficiency of B12 because it turns out we have like 3 to 5 years of stores of B12 as opposed to an order of months for folate. It's very easy to get a dietary deficiency of folate, but it's much more difficult to get a B12 deficiency. However, if someone becomes a vegan and is having no animal products and no dietary source of B12, eventually they'll run out of B12. But it does take a while. So you could have a dietary deficiency, but the most common cause is malabsorption of B12 through a mechanism of an autoimmune process affecting intrinsic factor from the parietal cell and lack of vitamin B12 absorption from the terminal ileum. So you could have malabsorption, which is a much more common scenario. And in that case, if you have B12 deficiency, you see that you end up with a problem. You end up with trapping this N5-methyl tetrahydrofolate that never gets back into the cycle. Okay? So a B12 deficiency causes a trapping of the folate in this form and functionally depletes this cycle, causing virtually the same effects as if you had a folate deficiency in terms of DNA synthesis. Okay? So this picture is really important because what it shows is that if you're folate deficient, you can give them more folate and you'll replace the folate cycle. Okay? If you're folate deficient, giving B12 isn't going to do you any good, right? If you're folate deficient, you need folate to get the green pathway going. Now, on the other hand, if you're B12 deficient, and the problem is that you've trapped your folate as N5-methyl tetrahydrofolate, and you've depleted this this green pathway, you don't have any more of this going around. If you resupply and give enough folic acid, folate, to someone who's B12 deficient, you can get this cycle going again and you can start to recover your DNA synthesis, and the anemia hematological findings go back to normal. Now, the problem with that is that B12 isn't just involved in DNA synthesis. It turns out that it has some other functions. And for B12, one of the other things that it does is that it's involved in odd chain fatty acid oxidation and metabolism. So even chain fatty acid oxidation occurs in a very similar pathway until you get this molecule propionyl-CoA, which needs to do an alternative pathway, and the step to methylmalonic acid occurs. It's different. You form this intermediate, which you don't see in even chain fatty acid oxidation, and then in order to get this to be fully converted to your succinyl-CoA and go off into TCA cycle or whatever, that's a B12 dependent enzymatic process. Okay, so B12 is also involved in other things. Okay, now some people believe that this is the pathway that's responsible for the problems that you see with B12 deficiency with respect to the neurological system, the nervous system. Okay, you get a lot of selective vulnerabilities in the nervous system for B12 deficiency. There's damage to peripheral nerves, optic nerves, some of the spinal cord white matter tracts, and people believe that if you're B12 deficient, you start to incorporate some of these molecules into the myelin and they cause it to be dysfunctional. That's probably not true. This is probably not the mechanism, but it's just helpful to remember that a B12 has an extra thing that it does, and that helps me remember that it can affect the nervous system. And this pathway is unique to B12. Okay, so this is an important consideration. Now, if we go back to our B12 and folate cycle, another consideration is that B12 and folate deficiency, like I said, are the most common causes of megaloblastic anemia, but there are other ways you can get it. And hopefully this diagram helps you see that. For example, there's chemotherapeutic agents that affect DNA synthesis. Okay, and there's a lot of these things. You have pyrimidine analogs like 5-fluorouracil. You have drugs that can block this enzyme, for example. You have methotrexate with the antifolate drugs that block this enzyme, right? That would cause a similar scenario, right? It would deplete this cycle. You have other drugs that mess up DNA in other ways, that there's other pathways. You have drugs for HIV, some of these antiviral nucleoside analogs. There's other drugs that block ribonucleotide reductase and affects DNA synthesis in other parts of the pathway. And so there's a lot of different mechanisms, but the underlying principle is that if you mess up this cycle or DNA synthesis in any way, you end up with a problem with those red blood cell precursors not going through the normal maturation process. And so to summarize what happens then when you have a problem with the DNA, we can make a list. And so the first thing that we said is that you're going to have anemia, of course. That's sort of the first thing that would come to mind. Okay, and so we can call this number 1. Okay, you obviously have anemia. Okay, and if you think about how you classify anemias, a very good model to do it is to first look at the reticulocyte production index, which was described in another video, and usually your RPI, the reticulocyte production index, is less than 2, less than or maybe equal to 2, which indicates that there's a decrease with production. Okay, and this ineffective hematopoiesis is under that category. All right. Now, what else would you see? You would see macrocytosis, right? Now, what's the best way to figure that out? Well, you look at the MCV, the mean corpuscular volume, and usually this is like something greater than the normal. High in the normal is 100 femtoliters. These usually a little bit bigger than that, maybe something like 110 for the MCV. And that indicates that the red blood cells are large and too big. Now, you also can get some other problems. DNA is important for other cell lines. For example, white blood cells and platelets also require DNA to go through their maturation process, and so you might get a little bit of a mild pancytopenia, which means that you have all the cell lines are down. This isn't going to be as profound as aplastic anemia, of course, but this just tells you that other cell lines are affected. It's not limited to the red blood cell. Now, another thing that's going to happen is, and it's not clear exactly why, is that your neutrophils that normally have 3 to 5 lobes commonly end up with greater than 5 lobes, and they call this hypersegmented neutrophils. Okay, hypersegmented neutrophils, and you can see this on a peripheral blood smear. All right, and this is very classic for megaloblastic anemia. Okay, and then if you wanted to think about other sorts of biochemical changes, you could think of number 5. Homocysteine, like we looked at in that metabolic pathway, homocysteine should build up, so you can get an increase in the levels of homocysteine. Okay, something like that. And because that there's a maturation defect, when we discuss that there's going to be some intramedullary hemolysis, you're going to get a little bit of an increase in hemolytic products in the blood. You might get a little bit of increase in lactate dehydrogenase. You also might see an increase in unconjugated or indirect bilirubin. These are just indicators of some hemolysis. There's probably a little bit of hemolysis outside the marrow. You know, these large cells probably have a little bit of reduced lifespan anyway. But the idea is that this is telling you you're getting some destruction of those abnormal cells as well. All right. Now, all of these are features of megaloblastic anemia. And we said that B12 is special and different because it does some other stuff. Okay. So when you have a B12 deficiency, all right, and we're going to go back to our picture over here. When you have a B12 deficiency, there's a couple of other things that you get. Okay. So we'll say B12 deficiency. And we're just going to put it here because this is going to help us remember. You get a couple of other things because you have B12 involved in this pathway here. You get an increase in MMA. All right. And we said that you also get some neurological changes, neurological damage to selectively vulnerable places in the nervous system. And like I said, the peripheral nerves are selectively vulnerable, so you can get polyneuropathies. Usually they're sensory. There's certain vulnerable regions of the spinal cord, the dorsal columns, and the lateral corticospinal tracts, particularly and collectively, sometimes people refer to that as subacute combined degeneration, optic nerves, and also some other places of the cerebral cortex. So you could see you'd start to get a variety of different neurological findings like paresthesias, tingling. You can get loss of proprioception vibration sense, some weakness, also some vision loss, and some neuropsychiatric changes. So all of these things are specific for B12 deficiency. You don't see that in folate deficiency or any of the other megaloblastic anemias. All right. So it's important to remember that, because if we go back to our picture of how all this works, we said that if someone was B12 deficient and you gave them folate, yes, you'd correct their anemia, right? And you'd probably be very, very pleased that their hematological changes were improving. But if you just gave them folate and you didn't correctly identify it as a B12 deficiency, you would not stop the neurological damage, and then that would be a major medical error. So it's very important when B12 and folate deficiency are on your differential diagnosis that you tease out and figure out which one of those it really is. And like I said, the most common cause of B12 deficiency is something called pernicious anemia, where there's an autoimmune process. So there's ways that you can figure out what's going on. You can measure vitamin B12 in the blood. You can look for antibodies for intrinsic factor. You can even measure folate, folate levels, red blood cell folate levels, and there's a variety of different things you can do to kind of figure this out, but it's desperately important. And also, I think it's good to remember that in addition to folate and B12 deficiency, which are by far the most common causes of megaloblastic anemia, there's other ways you can get it indicated here on this diagram. There's drugs, chemotherapeutic agents that target some of these enzymatic processes, or other drugs that affect other steps in the DNA synthesis pathway. And I'll just mention one other drug because it's kind of interesting, and that's nitrous oxide. Okay, and nitrous oxide, it's not totally clear how it works to cause megaloblastic anemia, but it has an effect on B12, and some people believe that nitrous oxide might be blocking this process here. I'll put N2O. And the reason why this is important is that when you have nitrous oxide blocking the effects of B12, you can start to get rapid deterioration and development of megaloblastic anemia when it normally is a generally slow process. I mean, we said B12 takes a while to become deficient. Repeated exposure to something like nitrous oxide can cause something called an acute megaloblastic crisis, and it's the most common cause for that. So people need to be real careful getting exposure to this, because it not only causes the megaloblastic changes, but also causes the neurological changes as well, because it's affecting B12 here. So hopefully now you have a good idea about megaloblastic anemia and the overarching mechanism of decreased DNA, ineffective hematopoiesis, a variety of important features, and the vitamin B12 and folate cycle and biochemistry that explains a lot of the findings. And that concludes this lecture on megaloblastic anemia and vitamin B12 folate biochemistry.